Hello and welcome to the program. I'm your host, Neil Howard, here on Health Professional Radio, and I'm real glad that you could join us once again. Our guest is Mr. Himanshu Brahmbhatt. He's the co-CEO of EnGeneIC. It's a biopharma company, and he's joining us on the program today to talk about the company's development of a first-in-class Cyto-Immunotherapy platform that's targeted for delivery of chemotherapeutics directly into tumors. But I'll let Himanshu tell us all about himself briefly and all about his company's development of this groundbreaking technology. Thanks for joining us on the program.
Thank you very much, Neil, for this opportunity. So, EnGeneIC, as you mentioned, is a clinical stage biopharmaceutical company based in Sydney, and we also have a U.S. subsidiary with an office in New York, mainly to carry out the clinical trials in the United States. Along with Dr. Jennifer McDermott, who is the joint CEO of EnGeneIC, we are the sole inventors of this entire EDV technology, with now almost over 410 patents granted worldwide. And the company is currently aiming to list on NASDAQ over the next year or two. Essentially, what we discovered was bacteria can produce little nanocells, and this is just a random event that occurs in nature. And when we engineered these bacteria, what we could do is in the manufacturing, we could actually reliably produce these little nanocells. They're non-living nanocells, and they could be produced in very large numbers in the laboratory and in manufacturing. Now, we then discovered that you can actually load these nanocells with chemotherapeutic drugs, and these drugs do not leak out of these nanocells. And since they are made from bacteria, on the surface of these nanocells, you have little carbohydrate structures. So we can attach bispecific antibodies where one arm of the antibody can link onto the EDV surface and the other arm can be targeted towards a cancer cell surface receptor. Now, when we inject this three-component system, which is antibody-targeted and drug-loaded nanocell, into the blood system of a cancer patient, these particles are 400 nanometers in size. So they are too large to get out of the normal blood circulation. So they are retained in the blood circulation. But it has been known for a long time that wherever there's a tumor growing, the blood vessels are highly defective and very leaky. So these particles rapidly drop out into the tumor environment, and then because they have the targeting antibody, they lock onto the tumor cell surface, and the cancer cell automatically swallows. It's like a Trojan horse. It swallows the EDV particle. We call this EnGeneIC Dream Vector. It swallows the nanocell, destroys the nanocell, and the drug payload is delivered directly inside the cancer cell. So in our patients, with almost 1,000 doses now delivered in patients, we have seen hardly any toxicity. It goes straight into the cancer cell and knocks them off.
So none of the healthy cells that are surrounding that tumor are affected due to leakage that was previously present in other types of or similar attempts at this method of delivery?
So previous methods that people have used, like synthetic nanoparticles, unfortunately, synthetic nanoparticles tend to leak the drug payload. So therefore, the drug still gets out into the normal tissue and they still experience toxicity. And the other thing is that all these synthetic nanoparticles are too tiny. So they actually can also get out of the normal blood vessel system. Our particle is too big. It's like a giant football, and it just gets retained in the blood vessel system and just doesn't see normal cells. And so this is the big difference. So essentially, size does matter.
What types of tumors is this new technology effective on? Is it all types of cancers across the board, or specific types?
So most solid tumors. And we have now, that's why, our clinical trial designs are very unique. When our clinical trials are done, people say, okay, I'm doing a clinical trial, say, in pancreatic cancer or brain cancer. We have all comers because this nanocell can carry a highly toxic payload, and wherever there's leaky blood vessels surrounding a tumor, it gets in there and it knocks off those tumor cells. So we've seen dramatic anti-tumor efficacy results in brain cancer in the United States, also in pancreatic cancer, in bladder cancer, in mesothelioma, in lung cancer, and these are all end-stage cancers where patients have run out of treatment options. So it's going into a lot of different cancers.
What type of extra longevity are patients seeing after this treatment?
So in the case of mesothelioma, we saw median overall survival in 27 patients jumped up to 41 weeks, and that was the last follow-up. Now, normally at the end stage, when these patients are on oxygen, their lungs are filling up with liquid, they're being drained constantly, these patients normally would survive 4 to 6 weeks. And instead, ours jumped up to 41 weeks at last follow-up. In lung cancer, we have seen patients live an extra 2 years. In New York, where we've got this dramatic result in a brain cancer patient, normally again, 6 weeks would be maximum at that stage. And yet, this patient is still alive after 2 years and completely stable. So we're seeing, you see, if the tumor gets into remission, the longevity can be dramatically enhanced. People can go on. Minimum what we are seeing is stable disease with prolonged overall survival and minimal to no toxicity.
Now, I understand that, you say you have over 200 granted patents worldwide. You've also developed a second generation EDV as well. How is it different from the one that we've been talking about in our conversation thus far?
Yeah. So in the first generation EDVs, we were only packaging conventional chemotherapy drugs into the EDV and targeting the EDVs using these antibodies on the surface. And, when we saw these dramatic anti-tumor responses, along with the co-founder, Jennifer, and myself, what we thought is, okay, one patient out of, 10 did dramatically well, and the others got, stable disease. Mm-hmm. Wouldn't it be wonderful if 9 out of 10 patients did very well? So we tried to analyze what was so different, where a patient derived a dramatic anti-tumor response compared to others who got just stable disease. And what we observed was truly remarkable, that in those patients where you saw a dramatic result, the patient's immune system had kicked in. Mm-hmm. So it was not just about killing cancer cells. If you wake up the immune system in these cancer patients, that immune system is so powerful, it can virtually wipe out most of the tumor. So then we thought, okay, how can we actually do this more reliably in most of the patients? So what we found is that we could actually take another EDV particle, another nanocell, and package it with the immune-stimulating molecule. Now we combine the two EDVs together, two nanocells, one carrying the drug and targeted to the tumor to kill cancer cells, and the other EDV is an immune stimulator. And when you combine these two together and you inject it simultaneously into cancer patients, both EDVs go and do a different job. One to kill cancer cells directly and the other one to wake up the immune system. And this is what we are now calling the second generation EDV, and that's what's going into these phase 2 trials in several cancers.
How accessible is this technology going to be in the end stages as far as cost of goods? In wrapping up, let's talk about how what you see as the future of these EDVs as far as the cost.
So this is a very interesting question because, most people are now aware that these new cancer therapies that are coming out from major pharmaceutical companies, they can cost anywhere from $100,000 to $800,000 per patient per year. Now, this is a tremendous burden on healthcare reimbursement agencies and affordable only in the most affluent countries. Needless to say, poorer countries, people just cannot afford these kinds of treatments. The great advantage of this EDV therapeutic is that the cost of manufacturing is remarkably low. To the extent that, at the moment when we manufacture these EDVs and send it off to the hospitals, a patient dose costs us only $200 to make. So you can imagine that what we want to do is that make it affordable worldwide. It shouldn't be a drain on reimbursement agencies. And, let's take the greed out of all this kind of stuff. This is about people's life and suffering, and we have to do something. And so this is one of the great advantages of this technology that, since it's bacterial in origin, you can just manufacture it very cheaply.
But where can our listeners go online and get some more information about your company, EnGeneIC, and also about your EDVs, your EnGeneIC Dream Vector?
Thank you. So on the website, most of the details are there.
And the name of the website,
So www.EnGeneIC, EnGeneIC.com.
Himanshu, thank you so much for joining us on Health Professional Radio. It's been a pleasure speaking with you.
Thank you very much, Neil.
You've been listening to Health Professional Radio. I'm your host, Neil Howard. Transcripts and audio of this program are available at hpr.fm and healthprofessionalradio.com.au. Be sure and visit our affiliates page when you visit our platform. You can also listen in and download at SoundCloud.
Thank you for listening to Health Professional Radio. We're very proud to be an independent broadcaster providing our content free of charge to you, the listener. One of the ways that we're able to remain free and independent is by having people like you become patrons. You can support Health Professional Radio simply by visiting hpr.fm and clicking the button that says Become a Patron. Your patronage of even just $1 a month lets us know that you're there, which in turn makes us more valuable to advertisers. And, of course, if you're able to afford more, then we would certainly appreciate the support. My name is Toby Longhurst from Health Professional Radio. Please visit hpr.fm, click the Become a Patron button, and support us if you can.